In this video on DNA cloning, I'm going to talk about cell-based in vivo DNA cloning and cell-free in vitro DNA cloning. In vivo and in vitro are terms that you will often hear used, and in vivo means taking place in a living organism, and in vitro means taking place outside a living organism. Basically, in vitro means taking place in a test tube or dish in the lab. Initially, DNA cloning was pretty much an in vivo exercise, but now it's a mixture of in vivo and in vitro. So, what is DNA cloning? Well, it's just a method of selectively amplifying DNA sequences to generate homogeneous DNA populations. And we can do this by either introducing the DNA to cells such as a bacteria or yeast and then growing up millions and millions of the cells and therefore producing millions and millions of copies of the DNA, or we can use the polymerase chain reaction, PCR, to make the copies. So, why might you want to make all these copies of a piece of DNA? Well, you may wish to clone the DNA so that you can explore some aspect of a disease or explore the function of a gene or protein in a cell. Or you may wish to clone the DNA so you can make the protein in the lab for further study. There are a large number of reasons as to why you might want to clone a piece of DNA. In terms of the DNA we can use, there are two different types that we generally use in the lab. We can use genomic DNA or complementary DNA, also called cDNA. Genomic DNA is the DNA from the genome of the cell, whereas the cDNA is made by using an enzyme called reverse transcriptase to make a cDNA copy of a messenger RNA. You must understand the difference between these two different types of DNA, because there is no point using genomic DNA if you want to make a protein, as it will contain both exons and introns. Likewise, there may be no point in using cDNA if you want to study some aspect of DNA transcription and regulation. For cell-based in vivo cloning, there are four key steps. 1. Cutting the target DNA from the original DNA source. 2. Inserting the target DNA into a plasmid, and a plasmid is just a circular piece of DNA that has properties that makes it useful for cloning work. 3. Introducing the plasmid into a bacteria or yeast, and 4. Identifying the bacteria or yeast containing the plasmid with the inserted DNA. Two essential tools we use in DNA cloning are restriction enzymes and plasmids. Without restriction enzymes and plasmids, molecular biology and the process of cloning wouldn't be possible. A restriction enzyme is an enzyme that can cut DNA at specific recognition sites, that is, at specific DNA sequences. When the enzyme cuts the DNA, it can produce either sticky or blunt ends. A blunt end, as the name suggests, is a cut straight across the DNA sequence, whereas a sticky end has an overhang, and you can see these two different types of cut in the image to my left. The upper image has a blunt cut and the lower a sticky end cut. A plasmid is just a circular piece of DNA that has some properties that means it can be used for cloning. These properties may be things like genes encoding antibiotic resistance or some other type of marker that allows us to detect the plasmid in cells. It may be a site that allows replication of the plasmid, a site that contains multiple restriction enzyme sites that can be used to cut the plasmid, and the plasmid may also contain sequences that allow it to express a protein from the inserted DNA. In the lab, we would cut the plasmid using one and often two different restriction enzymes, and then remove the small section of DNA we have cut out. If we use two restriction enzymes that produce two different sticky ends, then the two cut ends of the plasmid will not be able to join back up with each other. The DNA that we want to insert into the plasmid will be cut using the same enzymes that we use to cut the plasmid. The insert will then be able to fill in the cut region of the plasmid, and if we use two different restriction enzymes, we can even have control over the final orientation of the insert in the plasmid. The insert is then sealed into place using the DNA repair enzyme DNA ligase. To get the plasmid into the bacteria, a process which is called transformation, we can use one of two methods: a chemical transformation that often includes a heat shock step, or electroporation, which is when we use electricity to punch holes in the membrane so the plasmid can diffuse in. Okay, So that's how we would clone a piece of DNA into a bacteria in the lab using in vivo techniques. Okay, so in this video we've looked at how we can clone DNA using in vivo and in vitro methods. Usually in the lab we will use a combination of these two approaches. You may start off using the in vitro methods such as PCR to get the DNA you need, but then switch to the in vivo methods to complete the work.